while you're firing up the computers, I'm going to introduce this project. So in years before, we don't do this exploratory project. We just do the regular labs like mineral nutrition and water potential and things like that. But we decided to try something new, and according to the first section of the students, they say I think it was Dalton. He said he feel like a scientist after doing this. So hopefully you will feel the same. So in this part, each group is going to be assigned one Arabidopsis mutant. It's an insertion mutant we ordered from the Salk Institute. So for insert mutant, it means a tDNA or a DNA sequence is inserted by scientists into the plant gene, and so it disrupts the gene and makes it no longer functioning. So that's why we call it a mutant. So it's not functioning in the gene that's disrupted by the insertion. And each group will have your own gene, and we've planted the plants for you. You will do the online analysis this week, and after tonight, you're supposed to come up with a hypothesis of the function of the mutated gene. Next week, we will give you the plants. You will grind them up and extract DNA from them. And so that's for genotyping. And we are going to do PCR reaction on the DNA you extracted from the plants. So PCR, it stands for polymerase chain reaction. That's the experiment where you amplify the gene. And I will explain about it later in detail. So after genotyping, you're going to observe the plant for some time and see if your hypothesis about the gene function is correct. So that gene is mutated, which means your plant is going to show a phenotype or deficiency in the related function of that gene. And you will compare it against the wild types so that you know in which way it's deficient. So do you have a general picture of what we are doing?
Mm-hmm.
And this project is going to be spread out throughout the semester. It's going to take a few weeks, but as long as you follow the steps, it's going to be fine. It may sound less familiar to you because there's a lot of jargons, just like the paper you read and discussed with Dr. Hauser, but we'll walk you through it, so don't worry too much about it. Okay? And if it was me and you.
Oh.
But if I ruin it, don't blame me.
Okay.
I'm trying my best.
Are you guys able to connect to the internet?
Yeah, Jane fixed it.
It's funny when you find yourself looking from the outside. I'm standing here, but all I want is to be over there. Why did I let myself believe miracles could happen? 'Cause now I have to pretend that I don't really care. I thought you were my fairy tale, a dream when I'm not sleeping. A wish upon a star was coming true. But everybody I knew the melody that I heard you singing. And when you smile, you make me feel like I could sing along. But then you went and changed the world. Now my heart is empty. I'm only left with used to be's and once upon a song. Now I know you're not a fairy tale, and dreams were meant for sleeping, and wishes on the star just don't come true. 'Cause now even I can tell that I confused my feelings with the truth. Because I like the view when there was me and you. I can't believe that I could be so blind. It's like you were floating while I was falling, and I didn't mind. Because I like the view. I thought you felt it too when there was me and you.
Woo! Good job. Good job.
What high school music is that from?
I'm going to assign you your. So Christine, you're working with Melanie, right?
Yes.
Okay. Are both of your computers working?
Looks like I have it. It's Q4 Bioinformatics or Lab 4 Bioinformatics.
Okay.
Is that what I click on?
Bioinformatics analysis lessons, if you click.
Yeah.
Am I going under announcements or where am I going?
Announcements.
Okay, under announcements.
Are you video?
Yep.
Oh my gosh.
Oh, well, Jade's out of town.
Well, Jade's not here, so I want to make sure
okay.
she doesn't miss a single thing.
Oh my god.
You're going to be famous. You're going to be in High School Musical 4, Jane. I can tell already. There's been 3.
And which one do you want me to click on?
Are they doing 4?
Let's see your year.
Yeah. Really?
Yeah, there's mad links, like, which one are we going?
Like that. Like when is
Are you on
when is it
cameras
going to
now?
be? You want to work individually or work together? Like
Can I know?
Do you want to come back?
I mean, I don't know, man.
so. I need you to take down the number.
Okay.
So SALK
SALK
underscore
underscore
and 053778.
053778. Okay,
Yeah.
cool.
So this is going to stay with you
For forever. Keep for.
the SALK number I just gave you in the query box. Remember to include SALK underscore. So that's the whole name of the mutant line assigned to you. And hit search.
Hit search.
1.9.
Oh, 147.
Hit search. Click. Ta-da! I don't know what I'm looking at. La, 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 la,
Wait.
la, la, la.
So is the page refresh after
Yeah.
you search?
Yeah.
So among the mess of things, you should be able to find your number in like maybe a green rectangle.
Oh, I see it.
or-
Yeah.
Yeah.
It's in 2 different color triangle. It's like it's blue and green.
Let me see.
So like here's it right
Oh.
here, and then here. Well, this one's like 0.2130. So I guess we're doing the blue one.
Actually, we are doing the green one because that one also have SEQ after the like. See,
Okay.
SOC.
Oh, so like next sequence.
Yeah.
Okay.
And
Do I
yeah,
click it?
Yeah, click your. Click it. Yeah, click it.
There's nothing to do with it.
So in the there's one line labeled as note, and note.
Oh no.
And after it's another sequence starting with AT, is it?
Yeah.
So you also need to keep track of that number. So
Okay.
for Gabe's group, it should be AT5G41900, right?
Yeah.
For yours, I didn't know. I used theirs. So the AT stands for Arabidopsis cladiana. So it's like the plant's gene ID, the AT number. And the number before the G and after the T is indicating which chromosome the gene is on.
Neat.
And the last 5 digits are like just the ID number of the gene. This number will be very useful when you do information search.
Wait, you said the number like 5G, that's the chromosome you said?
So it's on 5th chromosome, and yeah. And the useful information here is when you look further down, there's one line with LOCN at the beginning. That means location. And after that, for your group, it's intron, right?
Yeah.
And what about yours? Is it
Core. Wait, are you asking what comes after?
UTR.
Oh, I'm sorry.
Yeah,
UTRS.
UTR.
Oh, 5? Or is that an S?
There are two, like UTR5 and
UTR5,
UTR5.
okay.
Like these are two different locations in exon. So for Cody's group, the insertion is inserted in the exon of the gene.
That's bad.
That's good.
But I
'cause-
mean, isn't the exon like the useful coding?
yeah, but
information?
we are trying to disrupt the gene.
Yeah.
It's a mutant.
But that poor plant.
Yeah, yeah, it's doomed.
Yeah. Anyway.
But if the gene is not an important gene, then it's fine. So, and for yours, the UTR means untranslated region, and the 5 means 5 prime, so it's upstream, upstream of the gene. And for your group, it's intron, so it's in an intron. It might or might not have symptoms, because insertions in introns may or may not disrupt the gene, but usually will. So that's the information you need to know from this page. Is everyone still with me?
Yeah.
Okay. So now we need to click on the second website I posted. It's also from the Salk Institute. So now we've come to the primer design. And as you know, primers are used for the PCR reactions.
Yeah.
Yeah. So, and it works like this. You have a none of the markers are really working well. So you have a double-stranded DNA.
Mhm.
When you heat it up, it's going to form a bubble, so open up in the middle. And the primers will anneal with the strands. And when you extend the primers, it's going to form a new strand of the DNA that's going to form the new DNA. So like half conservative replication, that's what it's called. So we're looking for the primers because next week we're going to extract plant DNA and do PCR, and you need to know where the primers come from. The primers we're getting today are actually, so remember I said one of the genes in your plant is mutated by an insertion. So suppose the rectangle represents the gene of interest, and like something is inserted into the middle of it. So it was originally like this long, but after insertion it's going to be much longer, right?
Is that, like, why is that? Is it because a new gene's being inserted, so it's just lengthening it?
The inserted sequence is not really a gene, but just any random sequence,
Okay.
just
Inserted in.
inserted into it. So
Okay.
it makes that gene longer. And when you amplify the gene's sequence, the original length may be a few hundred kilobases, but like after the insertion, it's going to be more than should the original length, I don't know, let's say it's 500 bases, but it may become 1,500 bases after the insertion.
Mhm.
So this marker is the PCR reaction. We are going to run the product in a gel, and you know that the longer the sequence, the slower it shifts because it's large and it's not
heavy.
moving that fast. So suppose this is our gel.
Mhm.
There are 3 situations. For the first one, you only have the slow moving strip, which means you only have the longer sequence. That's where you got a homozygous mutant, because both strands have been inserted and they are both longer than the original gene. That's the best scenario. The middle one should be a heterozygous mutant, because one strand is inserted with an outside sequence, the other strand is still normal. That's why you got 2 different lengths of the gene. For the last one, it's a wild type. Sometimes it happens because the insertion failed. So it's a fake mutant, it's not a mutant. Nothing is inserted and it's only got the original gene in its short sequence. So is this clear?
Yeah.
To we need the primers, and as to how we find the primer, you put the Salk number into the search box at the end of the page. And hit submit. We are leaving all the parameters as default because that usually works. And after you do the search, it's going to show up like a new window is going to show up. It's mostly blank with only the top line of information. You drag a little bit to your right, and when you find the 2 letters LP, have you found it?
Yeah.
And there's a string of characters right to it. Yeah, LP. Can you find it?
Yeah, LP, and it's a sequence, right?
Yeah, the sequence right to the right of it is the left primer. So LP stands for left primer. It's the primer on the left of the gene. And you can take that down. RP, that's the right primer. And there's also a sequence after that, and you can also take that down. To be extra careful not to make a mistake in the sequence, because otherwise it's not going to work properly. To 25 characters, and if one of them is mistaken, the temperature, the melting temperature is going to change, and it's not good for the PCR reaction. Website. And starting from the 3rd website down, you're using the AT number, no longer the Salk number. Okay? Open the 3rd web page. It's going to look like this, and by the name of it, it means the gene network of Arabidopsis. So ARA stands for Arabidopsis, and net means network, interaction. So what we're doing here is we put in our gene of interest, which is your mutant, and we look for the genes that's related to your gene, and we try to infer the function of your mutated gene from the genes that are related to the gene. Change the settings, just enter your AT number into the lower box. I usually sit, no, is showing up on the screen. It's got rank, so the rank is for the score that's next to it. The higher the score, the stronger the evidence.